Okay, I really like 1133. This is fun, and so we're not memorizing the structures of the nitrogenous bases anymore, but we are memorizing how the bases are attached to the 5-carbon sugar. We're memorizing a lot of the 5-carbon sugars, and this problem asks us to look at some of the different components, or ask us to analyze the 5-carbon sugars of a dinucleotide. So just a small chain of nucleotides, just 2 nucleotides attached together through a phosphodiester linkage. So let me diagram this for us right here. Switch to yellow. I think that's the easiest to see. So we have a phosphate here. I think that's a little too thick there. Okay, phosphate, and this is going to be attached to a 5' carbon. And then we have 4, 3, 2, 1, and then we have this tip of the house here, oxygen. And then this is going to be attached to a base. In this case, it's just adenine. Okay, so our next, this is going to be attached now through a phosphodiester linkage here. And I'm going to draw a little more detail than they have in the book. Five prime carbon here. 4, 3, 2, 1. Roof of the house there. And this is attached to a thymine. Okay. Now I'm not putting in all the extra hydrogens or anything. I'll put a hydroxyl down here. Why not? You know what? No, I better take that out because that's not in the book. We have a little arrow right here in the book. Okay, so this is the diagram essentially that the book gives us. And now what they're saying here is part A is asking us, is it DNA or RNA? Is it DNA or RNA? Well, they didn't diagram anything. They didn't put any OHs or H's here or anything, so we can't really use that. If there is an OH there, they left it out. But what can we say? Oh, we can look at this right here. Thymine, right? Okay, so thymine is found in DNA. It's not found in RNA. So okay, so part A, this is a DNA. These are deoxyribonucleotides. So in part B, they're saying, is the arrow closest to the 5' or the 3' end? So this arrow down here, is this closest to the 5' or 3' end of this dinucleotide? So this is the 5' carbon of the first nucleotide. So this little chain right here is going this way. 5' end is up here, 3' end is down here. So the arrow right here is closest to the 3' end. Okay, so arrow is closest to the 3' end. And part C is a little bit longer. So part C is, suppose that the molecule was cleaved with the enzyme spleen diesterase, which breaks the covalent bond connecting the phosphate to the 5' carbon. Okay, so that's a lot of information there. So we have an enzyme called spleen, you don't have to memorize this, spleen diesterase, and what it does is it chops covalent bonds connecting the phosphate to 5' carbons. That's what it does. Chops covalent bonds connecting phosphates to 5' carbons. Okay, so we're cleaving the bonds with spleen diesterase. After cleavage, to which nucleoside is the phosphate now attached, A or T? Okay, so we're going to cleave covalent bonds connecting the phosphate to the 5' carbon. Okay, so this is the phosphate we're talking about down here. We can chop this one too. But so we're going to chop this one. This is the 5' carbon. This is the phosphate. We're chopping this. We're chopping this covalent bond here. That's what that enzyme does. It's also going to chop this one. So the question is asking us to which nucleoside is this phosphate now attached. Now remember, a nucleoside is a 5 carbon sugar plus a nitrogenous base. So here's one nucleoside, 5 carbon sugar plus nitrogenous base, and here's the other nucleoside, 5 carbon sugar down here to a nitrogenous base. So which one is the phosphate attached to? It's attached to the adenine. The adenine nucleoside here. Okay, that's the answer to that one. Cool.